Vanakkam. Welcome to this course on classics in neuroscience. In this series of videos, we'll look at neural circuitry, specifically organization of the cortex as columns or cortical columns and cortical processing. In this video, we'll look at central visual processing and feature detectors within the visual system and the seminal, crucial Nobel Prize winning contributions of David Hubel and Torsten Wiesel. David Hubel was born in the United States in 1926 as the son of a chemical engineer. He mentions that he was born to parents who did not organize his life. That means he was given unorganized playtime. He could do whatever he wants with his time types, I suppose. This is something that is now recommended. Anyhow, he picked up hobbies ranging from chemistry, electronics, piano, Latin, a whole bunch of things where whatever he found interesting he pursued. He was also working in a farm, and he says something like he was working with parents who didn't organize his life, and it was much easier to reach the level of boredom necessary to think up things to do for himself. Something that's very interesting, something to keep in mind, because these are the kind of people who go on to become deep thinkers maybe. I think his family then moved to Canada, to Montreal, where he studied medicine and received medical training, and it was in 1950 he met with Wilder Penfield and Herbert Jasper, pioneers in those days in the field of neurophysiology, in general microelectrode recordings in particular. So he worked with Herbert Jasper in the EEG laboratory, EEG electroencephalogram, EEG laboratory in Montreal. That is in the Montreal Neurological Institute, that's where he worked. It was suggested to him, if he was interested in pursuing work in the field of neurophysiology, it was suggested to him that he better relocate to the United States, specifically to Johns Hopkins. But if he goes back to Johns Hopkins, or if he goes back to the United States, he has to work, I suppose, in the army or in the defense for some time. So he worked in the Army Research Institute. That research institute was called as the Walter Reed Army Institute of Research or somewhere, where he pursued similar line of work, and he found some interesting brilliant collaborators with whom he could work. For example, Michael Fouts. There he used small wire electrodes to make single unit recordings. Remember, what are single unit recordings? Those that record from extracellular matrix, from the outside the neuron, not intracellular, extracellular matrix. So this means that you are recording from somewhere within the near to the neuron, but you do not know exactly from where you recorded. Is it close to the cell body, or is it close to the axon, is it close to the dendrites, is something that you do not know. This is called a single unit recording. Remember, we mentioned this quite a few classes before, and he was instrumental in developing the tungsten electrode, which came to be used in many future experiments. Although it did not become famous at that time, a lot of people within the field of neurophysiology took notice and started using similar electrodes. And he also developed a chamber, because one of the concerns with recording from higher animals or mammals, for example, in this case the cat, is that there will be arterial and respiratory pulsations. To reduce that, he developed a chamber to close the opening the surgical opening that is performed. And he also invented a method to pass current through the electrode and to show the site of the recording. So, there are many things that he invented. These are all the experimental methods or the instrumentation or the experimental apparatuses that he developed or that he practically invented that helped him improve his own research output. And he started recording from the V1 or the primary visual cortex in response to flashes of light. And at some point, I think he became very frustrated that perhaps he was working for many hours and he found practically no response from a group of cells, that he was trying to identify what is happening. He was trying to wave his hand, and he found there was a grand response to his hand waving, and he found that some neurons responded to movement in one direction and another set of neurons responded to movement in a different direction. This was interesting because this means that there are neurons that are specialized to identify the direction of movement. It was among the first ones to identify this. It was a very critical outcome. He was very keen on pursuing the deep neurophysiology of this similar systems. He was advised to move to Johns Hopkins, and after some time he got in touch with Steve Kuffler. Vernon Mountcastle invited him to join Johns Hopkins Department of Physiology, where he started working with a new colleague, Torsten Wiesel. They were given a modest 225 square feet lab with a small budget. In today's world, your starting package when you start a new assistant professor research based university in a research division 1 research university, if you start, a new assistant professor would get upwards of 1000 square feet of laboratory space and at least $1 million as the startup, the seed grant, the first grant. This is this. So, in the good old days, they could do their Nobel Prize winning work with 1/4 of that space. So, remember, it is not the space or the equipment that does the research, it is the humans in the lab who do that research. Then comes the story of Torsten Wiesel. Torsten Wiesel was born in Uppsala in Sweden, where he grew up within the campus of a mental hospital that was headed by his father. So, perhaps it created some impression in him about the nature of the human mind. He was born in 1926 and he finished medical training in 1946 or 1947, around that time, and he started working with Carl Gustav Bernhard in Sweden. Bernhard was a neurophysiologist and he was working on several topics. Torsten Wiesel started working on the effect of local anesthetic on severe epilepsy, but somehow that work did not excite him too much. Later he tried his hand on child psychiatry, but somehow he was not particularly inclined to the detailed analytical nature of that work. Around that time, Bernhard informed him that one of his colleagues from America, Steve Kuffler, was looking for a postdoc, and Wiesel was already well read in this field and had read with much interest the work of Steve Kuffler and his colleagues in Johns Hopkins. So, it was very clear to him that when there was this opportunity available, Torsten Wiesel took over this opportunity and went to the United States. I think somewhere in 1954 or 1955 is when he landed in the United States, and he started collaborating with Steve Kuffler and his team. And his team was having many doyens, many pioneers in the field, such as Furspan, David Potter, Joseph Doodle, and Taro Furukawa from Japan. And initially, Torsten Wiesel worked with Kenneth Brown, on the cellular basis of electroretinogram. That is around the time, or just after that time, is when he met David Hubel. So, Steve Kuffler, being the extraordinary leader that he is, he brought both David Hubel and Torsten Wiesel together for lunch, and he suggested to them that perhaps they too can collaborate with each other, because David Hubel would bring his recording methods that he used to the visual cortex, to cortex in general, the visual cortex in particular, and Torsten Wiesel will bring his expertise in the field of receptive field and its properties. Yes. Together they can then build a theory of functional organization of the visual cortex. So, it's an extraordinary idea at that time. And remember that these two people come from slightly different backgrounds, and they bring complementary skills, and they were also getting along very well. They got along very well, so much that they started loving working with each other. Their model system, of course, was the standard model system of the time, which is the anesthetized cat, and they started recording from cells in the visual area or visual cortex with light that is shown on the retina. So, the stimulus was a physiological stimulus, not electrical stimulus. So, you shine light on the retina and then you record from various regions of the V1, primary visual cortex. Initially, the results were very disappointing. There was practically no progress for some time. Despite the initial disappointing results and the setbacks, the team continued pushing. This is important. It is crucial to have resilience. You need to keep pursuing, you need to keep trying different things. You need to, if something is not working, you need to quickly recognize and move on to trying other things. The results were disappointing, but they were also forming the foundation for the future progress that they were about to make. On one particular day, they were holding on to a particular cell for hour after hour. There was absolutely no response. But when they were moving the slide from one side to another side, the slide is going to dictate the type of stimulus that the retina will receive. That particular cell started firing, and it started firing a lot. Initially, it was not clear why is the cell firing, but later Hubel and Wiesel, they realized this cell had nothing to do with the specific stimulus, which was an opaque spot in that slide, but rather the fact that the slide was moving in and out. So, there was an edge that crossed from one side to another side. It was to that stimulus this cell was responding. So, this volley of response from that cell was obtained when the glass slide was moved slowly, because there was this fine moving shadow cast by the edge of this slide. So, if you move it fast, the response was not different, and if the orientation of the slide was changed, the response was different, slowed down, and at some point when the orientation was completely perpendicular to the original orientation, the response practically disappeared. This suggested that this cell was sensitive to orientations. These data they published in an article that they called as the receptive fields of single neurons in the cat's striate cortex. This paper on the receptive fields of single neurons in the cat's striate cortex was a landmark paper that opened a new field in the area of visual perception, and in the area of the functional organization of the visual cortex. It was a crucial paper in that field, and the figures were stunning with their clarity. Each figure told its own specific story. Also, they showed that specific cells responded to input coming from the specific eye, that is, there was ocular dominance. Whether the input is coming from left eye or right eye mattered. So, there was the first demonstration of ocular dominance, like you are having hand dominance, you are also having ocular dominance. These cells were driven by spots, and these cells were arranged not concentrically. Remember, when we discussed retinal cells, there was concentric arrangements and there was center out inhibition or center surround inhibition or excitation that was responsible for the retinal processing that we discussed while we discussed the retinal cells and the retinal neurons. Unlike the concentric arrangements of the retinal neurons, these cells were organized along a line that separated the inhibitory neurons and the excitatory neurons. In the retina, that was a concentric organization. Here, there was a line that separated the inhibitory neurons and the excitatory neurons. These cells were called as simple cells. These cells were tuned to a specific orientation. They were sensitive to movement in one direction. And of course, as I mentioned, that the kind of center surround inhibition or center surround excitation, this organization of center surround organization was not shown by these cells. The organization that we found in the retinal cells were different from these cells, and some of the cells, about half of the cells, responded to diffuse light. A critical reason as to why they were successful, why Hubel and Wiesel were successful, was because of the use of simple methods like spots and slits rather than using sophisticated instruments that could just send light at one particular wavelength, or very sophisticated instruments that could just send one very small amount of light in a particular wavelength, in particular intensity. Rather than that, the experimental method that was used by Hubel and Wiesel were very preliminary, very elementary, that just used simple setups, spots and slits. This really helped them study and explore different unpredicted response properties of the cells. Critical to note is that Hubel and Wiesel did not include latency as one of their measures. They paid no attention to latency. Remember, this was the age of Eccles, and his neurophysiology demanded measurements of latency. But we mentioned in our previous discussions that there are two approaches. One is to map out, one is to study circuits, and that required latency, that required stimulation and responses, electrical stimulation and response, and that. Two is to measure response of neurons to physiological stimuli. These are complementary approaches. One will identify circuits, the other will identify the set of all neurons, map out the set of all neurons that responded to specific types of stimuli, physiological stimuli. So, these are complementary approaches. They both can go hand in hand and improve our understanding of neurophysiology. So, that is what we discussed, and that is what has happened. In this case, Hubel and Wiesel were rather interested in studying response to stimuli more than studying latency and mapping out the exact or detailed circuitry that was involved. At that stage, this was their interest. Later, they pursued this line of work, and they also identified, like the simple cell, there were also some complex cells. These complex cells were not only like the simple cells in some sense, but they also were manifesting properties of selectivity. That is, they could selectively respond to specific stimuli. These results, all these results are the lore of visual neurophysiology. So, these results in the first 15 years after 1950, from 1950 onwards to 1960, or from 1956-57 when Torsten and Wiesel started this work till about 1965, are the grand old stories of neurophysiology, visual neurophysiology in this field. Critically, it is important to recognize the contribution of Steve Koffler in bringing Hubel and Wiesel together and pursue this line of work. It was also the age when, because he was the boss of the lab, practically in all the papers that came out from Hubel and Wiesel and others' work, he would have been the senior author and no one would have questioned. By the way, this kind of approach still continues in some places in the world, but this is not the way Steve ran his lab. People were free to choose what they wanted to do, and people were free to choose who they collaborated with, and people were free to choose what they published, and people were free to choose how they argued, took their publications. He just provided them space for growth. A fantastic approach that led to development of this kind of legendary science, science that later became legendary. Of course, in all the papers, because these papers were published in Journal of Physiology, we already discussed the authors were listed in alphabetical order. Because of that, it was always Hubel followed by Wiesel. It was always that because of alphabetical. This requirement later changed, and of course, women were identified just by their initials. Some things that we have now changed, but there are still many things that we need to change about this. Anyhow, so with this we come to the end of this video. In this video, we looked at central visual processing and feature detectors, visual feature detectors, and we looked at the stories of David Hubel and Torsten Wiesel and how they both came from slightly different backgrounds with complementary skills, came together and developed relatively novel methods for studying, simple but novel methods for studying visual processing, and came up with great theories of visual processing. Thank you for your attention.